Hello, everybody. My name is David Nakashi. I'm the CEO and co-founder of Protochips, and I want to thank you for joining us this afternoon or this morning, wherever you are, for our webinar on AXON. I realize most of you are probably joining us from home where you've been working for several weeks now, and I just want to say thank you very much for taking the time, and I hope all of you remain healthy and safe. If you were not able to join us last week for the webinar, our apologies. The attendance and the interest in this was way beyond what we thought, and we obviously didn't have enough seats available, and so we're running these this week. And we're very happy you could join us. Thank you so much. So, what I'd like to do is go over for a moment the agenda and what we'll be discussing and talking about today. Get this slide to advance. Here we go. So what we'll start off with is, first of all, a discussion on the product itself. The AXON is more than just a new product for Protochips. It represents a new vision for our company, and we'll spend just maybe a slide or two talking about what that means. It's going to be integral to the company. It's going to be core to the direction we're headed, and we can explain, most importantly, the architecture, because this first module and these first features really represent a first step in a new direction for Protochips that we think will benefit everybody, not only doing in situ microscopy, but many other regions, many other areas of microscopy in general. We want to get into some detail about how the software works. For those of you that may have had some introduction in the past, there were a lot of questions on what does the GUI look like, what options do I have, how does it work with the microscope, and we've got several video clips that show exactly what it's like to be sitting in the chair of a TEM and using AXON. We've got some examples of data and applications and a couple of new pieces of material that I think we're excited to show you. And in the end, we'll spend some time answering questions for you. Now, because it's a webinar, we're not going to be able to open the mics for everybody that's on here. But what you can see is over on the side, there's a questions box. And if throughout the webinar at any point in time, something comes to mind and you have a question, go ahead and type that in. And we have a moderator that will be looking for those. And if we have time at the very end, we'll try and answer them. And if we don't, we'll certainly get back to you just afterwards. But it's a great way to keep engaged. And please don't hesitate. Any question that you have in mind, just enter it in that box and we'll see if we can answer it. And then in the end, we've got a video and a survey. This video is something that our teams put together over the last month. We call it an experience video. And in today's day and age, when you may not be able to get into your lab, and certainly it's difficult for us to reach out and engage you directly, we took the time to make about a 5 or a 6 minute video all the way from front to back, loading a holder all the way to data analysis, to explain and show what it is like to use AXON, both for collecting and stabilizing your data all the way down to post-processing. And this is something I think you'll want to see. It's going to answer quite a few detailed and specific questions on the product. And then in the end, there's a survey. We'd love you to help us improve this to make it better. We intend to do more of these webinars to keep all of us engaged in this time. And also, if you're interested in understanding if your system is compatible with AXON, and for the most part, it probably is, but there are some caveats. That survey is a great place to get that information to us. Okay, so starting with the first point, where is the next frontier for EM? In other words, why Protochips? Where do we see this market going, and what can we do to help define that? I'm going to start with the end in mind. And if you take a look at this screenshot, this is what we call our notebook app. AXON is not just one single application, and Joe will get into an explanation on how all the products fold together. But this is the notebook app. This is the data analysis app, and it brings together the results of a lot of work that had been done previous to getting this point. So imagine if you've already done an in situ experiment or an experiment in general, and you've collected a large image stack, and you've exported this image stack and you're reviewing it in the notebook. If you look over on the left-hand side, you can see this is the series of images right here. In this case, it's a TEM image. You've got the X, Y, and Z coordinates of the stage, the time, and down in the lower left, you've got the temperature. This is for a heating experiment. And over on the right, you see the metadata. And this is all kinds of metadata. This is data taken from the microscope, data taken from the cameras, data taken from the in situ system, and it's all brought together. And when I play this video, and this is what happens and what it looks like when you scrub that image stack manually back and forth, you'll notice a couple of things. The first one is that particle, the large one in particular, isn't moving and drifting, even when you get to the part of the video where you're changing the temperature by over 100 degrees. The reason for that is it was drift corrected live, and you'll also notice that it's in focus. That's the first thing. The second thing you'll notice is all of the metadata changing. And what that means basically is that we have taken all the images, they've all been aligned and stacked, and they've all been brought into focus, and that was done live during the experiment. And each image has been stuffed with the metadata from the experiment. And what you're really looking at now, I think, is the power of seeing all of this brought together. You could imagine how much easier it is to draw some sort of conclusion based on an effect that occurs perhaps due to a temperature change or something else that you've set up. But this really represents a platform and a direction our company is going. It's not just about the capability of the holder or the capability of, say, some extra piece of equipment. What really matters and where we really want to make an impact is in the data itself to help you identify the key areas that you find are critical in an experiment and then consciously spend more time there. So why are we doing this? Why is Protochips making this software? Well, that's a question we get pretty regularly, and the answer is pretty simple. In situ microscopy adds a lot of new axes of data. It adds the element of time, the element of temperature, the element of, say, gas composition or flow rate or pressure, and all of these things just multiply the size of an already increasing data set. And we realize that what won't be helpful is to throw more in there. And as you probably know better than anybody, it is typically garbage in, garbage out. And so what we'd like to make sure we do is create a much higher quality of data and make it much easier to analyze and access for you. Second of all, if the quality of the data and the data synchronization isn't top-notch, scientific conclusions become difficult, if not impossible. And actually, even worse than that, you can draw the wrong conclusions. Cause and effect becomes difficult if everything isn't synchronized. And if something still has some movement or drift to it, it becomes difficult to see certain things in the data that otherwise would jump out at you if nothing was moving. So those things have to be addressed, and they're barriers for our market. In addition to that, as leaders in the market, we feel this is our place to apply our experience. We entered the market in the late 2000s with the first commercial MEMS chip, and since then, over the last 12 years, we've spent all of our time learning about all aspects of in situ microscopy, from catalysts to batteries to phase changes and all types of experiments through the years all over the world. And what we're continuing to hear regularly is these issues, these things that AXON are solving have been spoken to us for many years, and that product represents the culmination of all that experience and a solution for that being commercialized. Lastly, I'll mention this as I think it's important from a business perspective. AXON is a tool for everyone. It's not just for people in the Protochips universe. If you've got a holder that suffers from instabilities, whether it's an old heating holder that you have or a cooling holder or whatever it might be, and Joe's got some data he'll show you, you can use that with AXON, because our motto here is a rising tide raises all boats. We want to make the data generated in microscopy, and especially for in situ, more relevant to scientists in many new applications around the world. And that's an important mission for us. And so we made a decision to not just limit the use of AXON for our customers, but to make it available for anybody. Because at the end of the day, if the data you're generating is more compelling, if the data you're generating is a higher quality, then it's going to help all of us. It's going to help the market gain more attention. It's certainly going to help more people justify the time and the purchases for this, and it's going to help our business. So all of these things are very important. So a noble goal, and there's a lot of hopes and dreams, but several actual things have to be solved before we can get into that. The first one, again, is the data quality. And any of you who've done any sort of in situ experiment before, or actually microscopy period, if you've just put a cold holder into a microscope, you know drift is just something that you have to deal with. And typically the way you deal with it is time. You wait. In the case of in situ, that's a real problem because you are doing things in the experiment to cause these instabilities. And you can see in this clip, Yes, of course, MEMS technologies made stability and in situ just orders of magnitude above where they were 10 years ago. But you still have to wait sometimes for things to settle. And then what you really want to do is to be able to change the environment as you wish, and have those images stay completely still so you can focus on actually what's happening with a specific particle or a region of interest. And that's just not possible today. In addition to that, data exists in many different parts of the instrument. You've got data on the camera, perhaps on a GATAN computer. You've got data on the microscope, on a Joule or a thermo computer, and you've got in situ data on a Protochips computer. And they're in different locations, and it's really up to you right now to be able to bring all that together and synchronize it. And that usually involves a watch or an iPhone or a notebook and a pen, and that's hardly scientific or accurate enough. And then the issue, of course, is when it is all over the place, it makes interpretation difficult. It's hard to determine cause and effect. If everything is synchronized together and those images are stabilized, things begin to jump out at you that might have gone unseen before. And we know that's a pretty critical thing to solve as well. And so AXON really works to address all of these challenges. And so it also points where we're headed. So again, back to this quick video clip of the notebook. When all of those things are solved and they're all brought together, this is the result, and it is what you can see. And so where are we headed? We want to improve the quality of in situ data beyond just where physics can take us. So you reach a point where these MEMS chips took out a lot of material in mass from a heater, for example. They allow us to sandwich very thin layers of gases around a sample, gases and liquids, but you pretty quickly reach the limits of physics. There's no such thing as a perfect holder, and physically we're not going to be able to make one. But we realized several years ago that software and machine vision technologies can pick up where physics leaves off, and that really becomes a new sandbox of tools and technology for the company. It also brings together, we're looking to bring together data and streamline analysis. So again, once we get that solved, once we can improve the quality of the data, by bringing this all together in one place and providing a tool like this notebook to visualize and plot data, it certainly streamlines the process of analysis and makes it much easier for you to present very impactful publications, do data analysis, whatever your motivations might be. And that really is the end game, is the data. And lastly, workflows. You'll start to see us really get our head around new workflows. Workflows for EDS and analytical techniques, workflows to increase the throughput, workflows that decrease the learning curve and improve the safety. All of these things become, all of these are going to end up becoming new future directions for the company and for Protochips. So that's the introduction. And with that, I'd like to hand this over to Joe Sanguadolce. Joe's our Executive Vice President of Worldwide Sales and Marketing. And Joe's going to go through AXON in detail. He's going to explain to you what the product is, how it works, and what the context of that product is within the vision of our company to try and bring that all together for you. So again, I encourage you to type questions and put them in along the way. We'll be looking for those. So Joe, it's all yours.
Thanks, David. And thanks everybody for joining. So let's talk about why AXON. There are barriers to entry in the use of a TEM, even if you don't get into in situ work. TEM is a complicated technology. It takes some level of expertise and capability and experience to run one well. And so sometimes we see that one of the major barriers to researchers outside of TEM, some of the stuff that keeps them intimidated to dive into this kind of work, despite the benefits, is the fact that it looks hard to use and they would need to have support. When you turn your experiment into an in situ experiment, though, it becomes a lot worse. The whole point of in situ in many experiments is to incite bad behavior, to very aggressively heat and or place into a chemical, liquid, or gas environment a sample for the purpose of seeing how and if it changes or reacts. Those changes can usually lead to a great bit of instability. And so the sample will move around quite a bit. And so the work that's done in the microscope is typically done at lower magnifications. Therefore, you will not be accessing very high mags and high resolution data because the goal is to keep it at a low enough mag that you can never lose the area of interest that happens to be moving sometimes quite aggressively across the screen. And that's where the TEM operator comes in. They've got to constantly be repositioning, recentering the area of interest. And at the same time, since it doesn't move in just X and Y, it moves in 3 dimensions, the operator's got to simultaneously be maintaining focus of the area. And these experiments don't go for a fraction of a second or a few second exposure. Typically, they go for minutes and hours. Some are extremely lengthy as you exercise the entire reaction or the entire experiment. So this is a tedious operation that takes skill and muscle memory and can be quite daunting for somebody who's not used to it. But yet, many people do in situ experiments, and that community is growing constantly. So those that do the work generate a lot of data, and then they have another tedious task to try and publish that data, because it's not just taking the data and turning it into a movie. It's also aligning the parameters of the experiment accurately across that data. The data occurs across a timeline, and once you've married it to that, you also probably want to have different parameters of the experiment cut across that stack. And so you can see at some point in the imaging when something gets interesting or there's some anomalies or artifacts, you can tell what temperature you were exactly at that moment. And there are many other parameters in a typical experiment. Overlaying these and aligning this data manually is extremely difficult, and after you're done, you've achieved it with only a limited amount of accuracy. So the results can never be quick, and they can't be perfectly accurate necessarily. But this is the way it is. This is where we are right now. And so today, and for the past many years, the state of running an in situ experiment really isn't about the experiment itself. It's about operation of a transmission electron microscope. So in the design goals for AXON, the mission was pretty simple, to increase adoption of techniques like ours and to make things more appealing for people who want to transition into the use of TEM. We had to make the process easier. So we had to address the steep learning curve that exists right now in becoming proficient in the operation of a microscope. Hopefully, if we did this right, we were going to also open up some of the data in the midst of those transitions. Because again, if you're always running at low mag, chasing an area of interest around, you really can't focus on the experiment. And even if you try with a second pair of eyes in the room, you're going to see limited amounts of information because you're at low magnifications and things are changing. But we figured out that if we were able to stabilize things properly, there must be a lot of information inside of those transitions that, if able to be revealed, would be quite instrumental and beneficial to a lot of applications. Of course, if we did this right, all the experimental parameters, all the information about the experiment would be consolidated and married seamlessly and automatically with the data. And since this was always meant to be a software platform, it had to be something that had expandability. We had to be able to grow this so that once we established this platform and made this machine vision system work on your TEM, we could then roll out more and more exciting capabilities and features for different applications. So let's look at a typical experiment. This is today. This is 5 years ago. This is a typical experiment. We're heating a sample through about 50 degrees Celsius. This happens to be a nickel phosphide particle with silver in it. It's in a polycrystalline state. And as we heat it, it moves and it goes in and out of focus. So the operator would normally keep it at a very low mag, keep centering the area of interest, keep refocusing as best they could so we wouldn't lose it. And it doesn't look like there's very interesting information here anyway, but when you get to the very highest mag, if you pay attention, you'll see a couple of different directions of lattice information, which tells me the sample and the microscope are in a mode to deliver high resolution information. The problem is the instability keeps me from ever achieving that. So it's almost teasing me, saying, if you could control me, if I would behave, there might be a lot of interesting data available. What's the problem? The problem is simple. Your system is comprised of 3 major elements. It's the microscope itself, electromagnetically and mechanically. It's the imaging detector you happen to be using, whether it's a STEM detector or a digital TEM camera. And it is the sample in whatever holder it happens to be. And those 3 elements are critical to the operation. And yet those 3 elements are, to a large degree, disconnected. They don't communicate, they don't cooperate because they don't know when something that they see or that they do is right or wrong. So there's no ability to react. And so when the beam interaction generates thermal instabilities in the sample, the column doesn't understand that that's a problem or how to deal with that. And when the room environments change or modes are changed in the TEM and there's a long wait time for drift or stability to be returned, the sample can't comply and chase that along. So you're in the mode you're in because there are three major systems and they don't communicate. And that's where AXON comes in. AXON, as the name infers, is a connector. In the biological space, it's a connector between two neural cells. And in the example of the human brain, for example, those connections form a network that becomes an intelligent and cooperative scenario. And in the same way, AXON connects all these pieces of your system to cooperate and to become useful in an intelligent network that'll help us to achieve the goals of the design that I spec'd out. So let's go back to that experiment. On the left is the same thing we've been dealing with, what I showed you before. On the right is the dream that scientist would have if they had been able to make the sample behave. They would have probably chosen to go up on one of those particles, go up to a very high mag to fill the entire screen, watch that transition as the silver departs and the particle transitions from polycrystalline to monocrystalline in high resolution. And in fact, let's get rid of the distraction that the left side has become and let's just do our science. This is the dream that in situ experimenters would like to have. They can go to higher performance than they were ever capable of before, undistracted by the operation of the microscope, because to be honest, there is no one operating the microscope. AXON's taking care of it, and all I do is watch my science. It works on STEM detectors, it works on digital TEM camera systems. On that STEM image on the left, if you can see, you'll see little atoms of gold moving about in the matrix. On the right, the sample, as is typical of an in situ experiment, doesn't just move. This is the part where I try to address the comment that I get a lot, which is that this is just a drift correction system. Everybody's been using drift correction at some level on different systems for a long time. And drift correction in its simple state is just identifying and locking on an array of features with certain attributes or aspect ratios and tracking them as they move in space. This is not that. This is a very complex set of algorithms that understands fundamentally that the sample will probably not maintain its integrity. It'll change maybe violently over the course of a very short period of time. So our samples, when they react, they'll combine, they'll split apart, they'll change in contrast and phase, and they will morph in some astounding ways. And AXON's prepared for that. It locks on the background, it locks on the area I've asked it for, and it understands as it morphs that it is the area and it maintains it. So it's a very unique solution written by people who knew what to expect and who were trying to anticipate the kind of dynamic studies that our customers will do. This is the user interface for a Titan Themis TEM at North Carolina State University. And we installed AXON here. This is one of the development sites, actually, since it's local to our headquarters. For the last few years, we've had different flavors of the pre-production software loaded. And as I ramp up on the temperature that we've overlaid on this image, you see that the sample moves like you expect it to. It moves, it goes out of focus, and the operator is constantly pulling it back into the center and refocusing. That's the image on the left from the TEM. On the right is the AXON control screen. And we're booting up AXON. And since we're going to do a heating experiment, we're going to select the Fusion Select, Protochips Fusion Select electrothermal holder that we have here, start a session, and we're going to connect to the microscope. Once connected, the AXON screen delivers the exact same imaging as your main screen. So you can use AXON as your primary monitor if you choose. Those concentric boxes are there for capturing areas of interest for the drift correction to lock onto, and the inner box is something that's user definable if the customer wants to do focus assist and have it work on some features in what would otherwise be maybe a homogeneous area. So now we're heating the sample and it's drifting as we expected. We'll go up to a higher magnification and you can see the same thing we're used to seeing. But now if we want to stop that motion, we'll decide to reach over and click drift correct. And AXON now halts the motion. The AXON screen shows you a rock solid image, perfectly aligned. This will stay this way for as long as the experiment runs, and these experiments can run hours. But on the left side, you'll see the truth. The truth is that the sample continues to drift. In fact, at the bottom of the AXON screen, it's reporting over 220 nanometers per minute of drift on that sample. But AXON keeps pulling it back into the center of the screen, realigning it over and over and over, and it will do this for the entire length of the experiment. This is that notebook application that David mentioned, and this is a non-licensed part of the deliverable on AXON. So instead of doing all the work to publish and to scrub through your data on the microscope, you'll probably do this off at your office or any number of computers around your facility. If you have colleagues that collaborate with you and generate data on your microscope, they don't have to tie your TEM up either. They can take the data and they can install this software on their own systems and do the analysis and publication there. But this system allows you to do some pretty interesting work on the sample, and you can use it either live while the system's sitting there watching the data before you've actually captured a session, or after you've captured a session, you can use this system on the image stack that you've collected. Every moment, live or recording, this is gathering all the images with every piece of metadata that's associated with the microscope, the imaging detector, or the Protochips holder that you happen to be using. And I can scrub through this data and I can see that metadata change dynamically. I can put the data from any part of the system on the overlay image. I can put it up anywhere on the image I'd like, in any font size or font type. But once I get it set up, whether I'm live or it's a recorded stack, I can then scrub through and I can look for the kind of information that I want and maybe look for attributes or reactive quality of the experiment that would give me the idea that I want to publish that data. And once I get to an area where I find something I publish, I'm going to want to save that data. So I'll go ahead and hit the icon to save. The system will ask me where I want to save it, and I'll pick a target directory and name that. And then I can choose either the raw uncorrected data, the corrected, drift corrected data. I can have overlays on or off on any of it. And I can then choose to export the data to that directory. And once I've got it there, then I can open that data and I can use any standard software that I have, any custom software that I have. These images are not saved in a proprietary format. They're in lossless PNGs, which are industry standard. Every one of them has all the metadata embedded into it, so I can use standard image browsers or sophisticated image processors, whatever I choose to do the work I want to do. And also, when AXON exported the data, it didn't just send the image files there, it also consolidated all the metadata from every image into a singular CSV file that I can open, in this case in Microsoft Excel, and I can very quickly do some quick charting, and I can do something simple here like map the temperature ramp across time. But the idea is that there's a lot of aggregated data in the system that's captured automatically. And so you can use that to either visually graph or send to another system to do further analysis. But the data that's collected that represents the situation in all parts of your system is extremely valuable. And our customers are already getting extremely excited about this, because depending on what you're looking for out of your science, this can give you a wealth of different information. And AXON doesn't care what holder you used. It doesn't know what holder you used. This is a liquid cell, and in this liquid, the beam institutes a growth of gold dendrites on the extremities of these particles. And I'd love to watch that, but of course, when the beam starts that and then a few degrees of heat exacerbates it, it runs away from me and it goes out of focus very quickly. And this is typical. But with AXON on, I get to sit and focus on the area I'm interested in, watch the experiment unfold. In fact, let's get rid of the distraction of the old methodology on the left, and let's just go to high resolution and watch this thing unfold, watch my science with no one operating the TEM. This gas cell experiment doesn't look very interesting, but I've told AXON these are the areas I want to watch. And you don't realize this yet, but there's a very violent reaction going on all around these particles. In fact, the one on the left is about to be intruded by another particle that will try to consume it. AXON knows I wanted to stay here. AXON knows this thing's changing and morphing, and AXON will not allow me to lose my area throughout the entire experiment. So lots of people have MEMS-based in situ holders now, but everybody's got a bevy of holders in their lab, and I'm willing to bet that some, if not all of you, have a lot of these holders. Bulk heating furnaces, cooling and cryo transfer systems, tomography, strain, helium. There's so many different holders that people use that are all dynamic, all in situ at some level. And the problem with some of the purchases that people have made, this is an old story we see a lot. Many people have very high resolution microscopes. They buy a dynamic holder, a 1,000-degree heating holder or a liquid nitrogen cooling holder, and they figure they're going to be able to do now high resolution experiments at elevated or lowered temperatures. But when they marry the two technologies, the dream is cut short because the physics gets in the way. And when you start to heat these bulk samples, the instabilities induced by the marriage of heat in those holders makes it impossible to go to higher resolution or higher magnification. And so a lot of these holders don't get used in the way they were meant to or they were dreamt of when they were first purchased. And in fact, some of them just get put back up on the shelf and they don't get much use or no use for a long time. And that's terrible to have made an investment like that and not be getting the benefits of it. This is one of those holders on the left. It's a Gatan 652 tantalum bulk heating furnace. This is a very typical sample running this kind of holder. It's a 700 series stainless steel, electropolished. It's being ramped through a not very exciting range of temperature because this kind of a sample usually likes in excess of 600 degrees C. But even before we get anywhere near there, and even at these very low magnifications, you can see how unstable it is. It's moving in multiple directions, and there's no chance that I'm going to do any really high resolution work over the course of a real experiment. Turning AXON on makes it behave. It's in focus. It stays stable. If I go to higher magnification, these inclusions might have been interesting to study, but they're gone before I even get a chance to consider that. With AXON engaged, I can sit here all day long, going through the entire ramp of heat on this holder and never losing my area, enjoying the science, again, never touching the controls of the microscope. The unthinkable would be to go to high resolution on a sample like this. And just so we got the best possible result, the user, the owner of this holder who let us borrow it for this experiment, gave us some guidance. She said that normally in order to get some level of stability, they go to about 600 degrees C and they let it sit. So the sample and the holder can equalize at that temperature for an hour. After that hour, you start to heat and you see immediately with the first few degrees of heat, it takes off in a way that would make it impossible to get any real high resolution data. But with AXON on, I can sit and I can watch this at stunning high resolution for as long as I'd like, and I can just exercise the science on my sample. Again, never touching the controls of the microscope. It might not have been one of the primary design goals back in the day when this was first being envisioned. I'm sure it was more about trying to increase customers' use and productivity with our products. But in fact, AXON goes deeper in its correction than maybe initially planned. It fixes fundamental issues in the TEM system, and therefore it fixes and removes those instabilities from getting in the way of your use with even products that are not ours. It essentially takes all these holders that you spent good money on and may not get as much use as you'd like from, and it fixes them. It unlocks the potential, bringing them right back to where they were guaranteed to work. In fact, maybe orders of magnitude higher performance than they were ever guaranteed to work, because nobody would have envisioned that you'd have a perfectly stable environment to operate them in. This is a very routine sample or situation. This is a customer who does tilt work and has to go to certain zone axes on this strontium titanate and then get imaging. And it's something she does over and over and over. And she showed us one of the problems that she has, and that is that in this case, she tilted about 15 degrees to the zone axis that she wanted and then stopped tilting. And this is just an alpha tilt. It wasn't a double tilt, beta direction. But this movement you see is not tilting. It's the stabilization period that she has to wait for while this sample keeps on moving to a degree and she can't really get to work. But with AXON on, she can instantly go right to the work that she wanted. In fact, if I look at the data from these two image captures and sum them up, this is 275 frames of data summed from the uncorrected side and then the AXON corrected side. And you can see by the integrity of the AXON image that the alignment frame to frame was beautifully achieved, even on a very homogeneous type of image. So AXON, it's software, but more specifically, it's a software platform. This platform is fueled by software, and everything we've showed you today, everything we've talked about so far, and what the rest of this webinar will be, is about one piece of software, module number one, which is called Synchronicity. And Synchronicity does three main things. It has three pillars of delivery here. The first one is physical stabilization, physical synchronization of the sample in whatever holder it happens to be in with the microscope, with the imaging detector. The next one is parameter synchronization, taking all the data from the microscope, the detectors, the holders, and marrying it seamlessly and automatically with all the data that's being collected. And the third is experiment priority, synchronization of the parameters of the experiment so that not only can they be read and reported, if they're Protochip systems, you can actually turn it around and AXON can control the system so that it can maximize success in your experiment. And I'll show you that in just a second. So the physical synchronization, how does that work? It's basically like gluing your sample and your microscope and your detectors together in three-dimensional space so that relative to one another, they never move. And it's a very transparent layer of control. It's not interruptive, it's not disruptive. When you reach over to grab the controls on the TEM, there's no resistance, there's no pushback. It feels the exact same way as it did the day before the AXON was delivered. Because AXON's not taking over anything. It doesn't interrupt scans. It doesn't have a scan generator. It doesn't have any control interfaces that would disrupt your normal operation. So at any time, whether AXON is engaged or not, you can reach over and do anything you might do on your microscope without any change in the way it responds. And for example, if you change focus to a different depth of focus while AXON is engaged, when you're done, it'll say, okay, I guess that's the new focus depth that the scientist wants, and I'll respect that, and I'll keep on correcting, and I'll keep that focus. So it's extremely non-interruptive. It's not even as aggressive as something like the cruise control in your automobile. When you want to reposition a sample, physically or at least in the projection of the image to delivery to the detector, you do it with several different things. You can move the goniometer, which physically moves the sample. But it only really works it's a very coarse movement, so it only really works at lower magnifications. Well, you wouldn't use this at very, very high resolution conditions to touch up the position of a sample. You'd probably lose it. But then there are finer modes of operation like beam shift, where you're actually moving the projected image. And that's much better for higher resolution and higher magnification situations, but you've got to be judicious with the use of that, because if you use it too much, you'll induce aberrations into the electron beam that'll destroy your imaging quality. And then we're all getting very familiar with the kind of digital, either on the fly or post realignment, cross-correlative realignment that a lot of the newest cameras can deliver. And they're very effective. Unfortunately, they're only effective over a very small portion of a single frame of view. So it's impossible to have something like that be responsible to cover an entire in situ experiment, which could go on for hours and literally consume millimeters of travel physically on your sample. And so AXON has to make the decisions on when to use any or all of these things, but it does that seamlessly. It does it without your intervention. You don't have to set it in coarse mode now or fine mode later to do things. You never touch it at all, as a matter of fact. You just need to say, this is what I want to watch, and then you turn your attention to that. In the background, AXON does what it has to do, whether it can just get rid of a little bit of very small drift with digital means, or it has to actually change and move the goniometer over and over again. It'll take care of all of this in the background, and it will never violate the beam shift, moving too far out of center and giving you aberrations in your imaging. So parameters. Every moment as you're gathering the data, every one of those pieces of data has got all of the information from all aspects of the system embedded into it. And so if you've got a current day Protochips holder, you're getting all of the parameters from the gas cell, the liquid cell, or the electrothermal system, and not just the stimuli that you're sending into the sample, the responses you're getting back from them. So it's a wealth of information. But even if you're not a Protochips customer, even if you don't have Protochips holders in this experiment, you're also getting all of the parameters of setup on the microscope, KB, spot size, convergence angles, positions of the beam, of the stage, the images, what mode is the camera in? Is it binning? The refresh rates. All this information is there. All of it's being sent to every image without your decision in the way, and you'll use that data as you find occasion and opportunity later. And then experiment priorities. This is actually quite powerful. Just like on an SLR camera, if you were to be taking photographs of a high-speed event, a car race or a bicycle race or a sporting event, you might choose to have the camera help you by putting it in a semi-automatic mode of, say, shutter priority. And in that mode, the camera will be instructed to never allow the shutter speed to change, but to help you in achieving the best exposure for each of the images collected by taking control of and altering any other parameters that it has access to. AXON can do the same thing. AXON will be allowed to, for example, if field of view is the most important thing to your experiment, you never want to lose the field of view. If the experiment does something unexpected, which they can do, sometimes people will go halfway through or maybe even almost all the way through an experiment only to find that at one point the heat that's applied or the reactive conditions that are applied make the sample do something extremely aggressively, too aggressively to be not lost or not to destroy your experiment. And you'll have to go back and repeat that part of the experiment, or perhaps worst case, you'll have to go back and repeat the entire experiment. If you were to tell AXON that the field of view is something you never want to lose, and it was to see a reaction start to happen that was becoming more violent than it thought it could correct for, AXON could take advantage of some of the other parameters. For example, it could pause the temperature ramp or slow it down, and maintaining stability and keeping the movement within the parameters within the boundaries of AXON correction. So this means it ensures higher levels of success on all your experiments and a bit of automation to allow you to do the experiment better and get more out of it. With our products, AXON will also feed back to the Protochips holder system recommendations on settings. So for example, if it was that heating experiment that I showed you a little while ago, if we looked at the Fusion Select display, you would notice that it was also reporting a recommended maximum ramp rate. It's the AXON conclusion that that rate or slower would be a safe rate considering the way the sample was responding, so that we would never lose control of the sample. And AXON is a software system, but there's some hardware. So the piece of hardware that comes with it is called AXON Core. It's a hardware server, some speed NIC connectors and cabling, and the high resolution display. And we realize that most TEMs today have a lot of monitors and a lot of systems installed, and that the real estate on that countertop is extremely precious. So if there's room, you can put it there. If there's not room, it comes with all the hardware we need to mount the monitor for AXON above one of your primary monitors, wherever you choose. And so you'll always have AXON in a proper place so that you can use it frequently and with close attention. But after the hardware is installed and calibrated, it's all about the software. And again, everything we're looking at, everything we've discussed so far is about synchronicity. Synchronicity comes in 3 flavors: Pro, Lite, and X. The Pro and the Lite versions are for Protochips holder customers. You can use Pro if you have a newer Protochips holder, one with a current day operating system, and it will allow all of the features that I've shown: the physical synchronization of your system, the data synchronization from not just the microscope and the detectors, but also from the holders, the Protochips in situ holder systems. And because it's not just reading data, but it's controlling them as well, you'll also be able to do experiment priority. If you have an older legacy holder or something from us that doesn't have the latest operating system, and for whatever reason, if you decide you don't want to upgrade it at this point, you can use the Lite version. which will give you the physical synchronization, and it will still do the data sync, but only from the microscope and the imaging detectors. It won't know how to communicate with the older Protochips holder system. So you won't have that full functionality, and of course you won't have experiment priority. And then X is for everyone else. Customers who have no dynamic holders at all, no in situ holders, or who have holders made by our competition. They will also be able to enjoy the stabilization of their system, the increased productivity and the higher performance that physical sync brings. And they'll also be able to enjoy the data aggregation and analysis that's possible with the data sync coming from the microscope and from the imaging detectors. But obviously, we won't be able to talk to another brand or a different type of a holder for experiment priority or data sync of those parameters, at least not yet. After that, it's still software. It's module 2, 3, 4, 17. We've got a roadmap that's robust and extremely exciting, maybe for some of you more exciting than Synchronicity. And think about it, once we've established this machine vision platform on your microscope and stabilized the entire situation, think about what's possible by a system that's in full communication with all the components in the equation. It's pretty exciting. We are at a weird spot right now. Everybody is suffering with this crisis, and most of us have not been able to get into our labs. We've lost weeks, months of time, and I don't know for each of us how long this goes, but there's definitely some damage that extends past just delay in the deliverables for your experiments. And so when we come back to work, there may be financial damage. You may have lost some funding that was hopeful toward the purchase of different equipment, or things have been postponed or frozen. Your staffing may have been damaged at some level. But when we get back, even though we can't roll back the clock and reclaim all that instantly, I think AXON presents some very interesting options to our customers. That machine vision workflow system makes you far more productive. That means that whatever you would have done in those last months that you were off, you'll do in a fraction of the time. So you actually will be able to catch up a bit. And every piece of data that comes out of the system under AXON support will be theoretically at a much higher resolution, a much higher level of performance. So the work you do faster might actually be better work. And once you have caught up with the backlog of what you missed while we were paralyzed by this crisis, you'll continue to work with your TEM in what AXON will have established as the new normal of productivity, performance, and ease of use. When we marketed AXON, when we released it, we had to make another decision. That was how to price it. And if you check with your Protochips representative, you might be shocked to find out how inexpensive the system is. David mentioned earlier that Protochips is a leader in this market, that it basically invented the MEMS TEM in situ holder market. And if we considered ourselves the leaders, and if we really meant what he said about all boats being risen by a rising tide, then elevating the community would be the #1 goal. And as the leader, we decided to do exactly that. So we made the pricing of AXON such that most labs can afford it without a whole lot of resistance. So everyone should be able to take advantage of this. So in conclusion, this is a game changer. We're seeing it all around us. We're seeing it from the first users. We're seeing it from the experiments that we're running. It's been a fantastic rollout. And since the minute it was released in December, the excitement has been intoxicating. But it definitely has achieved the goals we were after. It's made TEM use of all kinds easier and more productive. But for in situ experiment scientists, you get to now enjoy the entire experiment, paying attention to your experiment and not being distracted by the need to use a TEM or to operate a TEM anymore. And while you're taking this data, you can do it at magnifications that you decide you want, but they're not limited anymore. And so we feel that through the transitions, you're going to find data, you're going to see information that was invisible before, and it should give a whole different value to the work that you produce. And all the while, all the data from everything in your system is being automatically aligned and inserted into the data you have. So publishing becomes easy. And in fact, the scope of that publishing is expanded past the imaging that you were after before. You'd be able to aggregate and to analyze that data and come up with a whole bunch of extra analyses and conclusions than you would have had access to before. And when you have our newer holders, you can also enjoy the control and the success that's bolstered by experiment priority. And remember, this is just the first software module. So David mentioned that there's a new movie. It's called AXON Experience. It should load when we end this webinar, but if it fails to for any reason, you'll probably be able to find it on our website. And also, I think Mike Coy, the Director of Global Applications Marketing, is going to send out information on how you can get to that. There's a survey, as David mentioned also. Take the time to fill that out. It won't take long at all. It's very simple. And it also gives you the opportunity to ask us for more attention. I want to invite everybody on this call. Please reach out to us through your local representative or through the website. Please let us know. We would love to schedule and hold a personal session with each of you and your colleagues and discuss this system and how it applies to you and how powerful it might be for your lab with each of you independently. We're all waiting to do this kind of work, and we do it a lot now, so don't be shy. So I want to thank everybody for attending. Jennifer McConnell is for Protochips, and she's been compiling questions in the background. And I think Jen is going to see if we can get through a bunch of these right now so we can answer some of them for you. Jen?
There you go. Thank you. Yes, we had a pretty decent handful of questions here, and I've tried to kind of consolidate them based on topics so we can cover as much as possible. So let's go ahead and get started. The first question I actually think is a good one for David. David, does AXON improve the data quality coming from other detectors like EDS and EELs, et cetera?
Thanks, Jen. Yes, we do get quite a few questions on EDS and EELs and other analytical techniques, as well as the distinction with TEM and STEM. So first of all, you saw data that Joe presented in the presentation, and AXON does take image input from both the STEM detector and of course the TEM camera and do drift correction and analysis on those. So that STEM detector, absolutely so. For EDS, obviously there's a lot of interest in this space, and what I would say is there are certain circumstances by which AXON can stabilize images for EDS. In general, though, this is going to be something that we'll really put our minds to over the course of the next whatever period of time here in the near future, and develop a module specific towards analytical capabilities. Just expect us to move in this direction before too long, because we realize its importance. We added analytical capabilities to our in situ holders over the course of the last 3 or 4 or 5 years, and look to us to strengthen the way that AXON is used with EDS in particular and other capabilities in the near term, in the near future.
Great, thank you. Joe, this might be a good one for you. How can I tell if my specific setup in my lab is compatible with AXON?
Sure. The simple answer is most TEMs are. But the specific answer is contact your Protochips representative, and they'll ask you some questions, and they'll check it out against the compatibility chart that the engineers have given us. And worst case, we can also have you install a small bit of software code on your system and run a quick check. But it's very simple and easy, and we actually have not run into a TEM, at least a Joel or Thermo TEM that we've met so far that hasn't been compatible.
Okay, great. Joe, this one is also for you. Don't go far. Where is the line drawn for quote unquote older versus newer Protochips holders for Synchronicity Pro compatibility? And can I upgrade the software later if I purchase the Lite or the X version?
Right. So older versus newer, the newer versions that have the newest Clarity operating system from Protochips are if it's a gas cell, it's the Atmosphere 210 series. If it's a liquid cell, it's Poseidon Select. And if it's electrothermal, it is Fusion Select. Anything older for Fusion, it would be just the regular Fusion or the Aduro line. Anything older than those 3 platforms, the 3 systems that I mentioned, would be considered legacy. But even if you don't have a Protochips holder, say you bought the X software, Synchronicity X, and then 6 months from now you decided you needed a MEM system or a gas cell, and you bought a Protochips system. When you got that, we would upgrade the X to a full Synchronicity Pro for the completion of that license term. So yes, of course, we'll upgrade you, and you can take advantage of Synchronicity to the fullest extent that your hardware allows.
Thank you. David, I think this next one is best for you. I think we touched on this, but what format is the data saved in, and can I use it with my typical post-processing software?
Yeah, so this was important to us as well when we talked about a rising tide raises all boats. There's a lot of options out there, not only with holders and systems for AXON to work on, but also from an image analysis standpoint. And we didn't want to get in the way of somebody's data analysis workflow. So we chose a lossless PNG format, and that's what you can export the complete AXON corrected data stack into, and that way you can bring those images into ImageJ, back into the notebook, anything along those lines. Any of the metadata that's written, obviously the metadata formats are unique to us, and those would be read out of the notebook app, but it's pretty easy to take information, data, images, and send them through that lossless PNG format to almost any type of analysis application you'd like to.
Great. I see we're running kind of short on time. Let's get to one more. David, can you give any ideas as to what the next modules will be?
Yeah, okay, so that's a great question. Obviously, we can't talk too much about our future roadmap, but I'd say maybe keep your eye on Protochips for a handful of things. One, this came up earlier, is a true analytical solution with AXON, and one that really, I think, addresses workflows in that space. We understand the importance, we're on it, and this is something that's near and dear to our hearts. A second one would be look for us to work quite closely with other types of workflows that help increase the throughput of data collection. I think there's obviously an appetite to do more types of statistical analysis and things like this. And I'd say the third one, and I think this started off as a project for us, but we realized the power of this in the beginning, and that's that notebook app. Look for us to continue to develop that notebook app in ways that allow the users to increase the type of analysis they do and the way they work with their data and filter data. We think that's going to be a pretty important part of our portfolio going forward.
Great, thank you so much. We're pretty much out of time here. Any other questions that haven't been answered, we'll come back and answer for you individually. But otherwise, I'll hand it over to Joe to wrap us up.
Thanks, Jen. Thanks, David. Again, we want to thank everybody. I know this is a challenging time for us all, and we want to thank you for taking time out of your schedules and day to join us for this. We're very excited about AXON, and we see the same level of excitement in the community. So once again, I'll invite you to contact us, schedule time with us to go into this at depth if you'd like. We love talking about it. We'd love to show you as much as you and your colleagues would like to know, so that you fully understand what we think is a real game-changing technology. Again, be safe, and we hope to see you face-to-face at some point soon. Take care.